Well, thank you for the introduction, and today I'm going to be talking about mRNA elements that are called riboswitches that are important for controlling gene expression. And so I'm going to start out by saying that bacteria must adapt to environmental changes very quickly. And one of the ways that they can do that we can actually see their phenotype through a microscope is they make biofilms. Biofilms are just proteins that certain bacteria excrete that form like a layer around a cluster of bacteria and protect them from certain environmental changes like the introduction of antibiotics or temperature changes within the environment. And that helps the bacteria to survive until the environment changes where they can continue to replicate. And for these environmental changes, for them to protect themselves from these environmental changes, they have to be able to regulate their genes, the expression of these proteins. They need to upregulate certain proteins, downregulate certain other proteins, and in bacteria, this can be done on mRNA at the level of transcription and translation. So originally, one way that bacteria can do this is on the level of translation. Bacteria have evolved certain regions in the 5' untranslated regions of protein-coding genes where repressor proteins can actually interact with certain regions of the mRNA and repress the expression of downstream genes that actually code for that repressor protein. And this is actually an example in ribosomal RNA where when there's an excess of ribosomal proteins, in excess of ribosomal RNA, the ribosomal proteins can actually bind to certain regions on the mRNA and repress the expression of genes that code for those ribosomal proteins. However, there were some systems that were regulating certain genes, but there were no protein repressors that were able to be found. This was the case for vitamin B12. When vitamin B12 was introduced to bacterial media, the genes that were, the proteins that were repressed in order to not have more vitamin B12 formed, they were being regulated, but there was no repressors that were found. And so this led to the idea that maybe mRNA elements were able to sense cellular metabolites on their own without having repressor proteins. This idea was put forth by the labs of Yevgeny Noodler and Ron Breker, and these RNAs were then termed riboswitches. Riboswitches are evolutionarily conserved, non-coding RNA elements located in the 5' untranslated region of, again, protein-coding genes. And the way they work is that they have two mutually exclusive conformations, a ligand-bound and a ligand-free conformation. When the cellular metabolite reaches a certain threshold in the bacterial cell, the ligand, the sensing platform of the riboswitch is able to sense this metabolite and bind it, change its conformation, and in this case, for example, this purple region is being sequestered by the specific fold of the riboswitch, and a terminator is formed, therefore no transcription can occur. However, when the threshold of the ligand goes down, the ligand unbinds from the riboswitch changes its conformation, and this region is now able to form the anti-terminator, and transcription can continue. This can also happen on the level of translation, since in bacteria, transcription and translation occur at the same time, and instead of this terminator or anti-terminator being formed, when the ligand binds, it sequesters the Shine-Delgarno sequence or allows it to be bound by the ribosome. And so riboswitches have been found in many pathogenic bacterial species, including Salmonella, Staphylococcus, and Bacillus anthracis. And they control very fundamental biological processes like toxic ion transport, cell wall metabolism, and amino acid and nucleic acid synthesis. And these are all, the idea is that these riboswitches can be targeted with drugs or compounds to be able to regulate the transcription or translation, or basically the expression of the downstream genes. Historically, riboswitches have been identified by bioinformatic means. And basically the way that people did this was looking at the untranslated region upstream of protein coding genes, grouping the sequences together using BLAST or later more sophisticated algorithms that can search for specific motifs, grouping these together, and then grouping the riboswitches based on the downstream genes that they're coding. There's been over 100 families of riboswitches that have been found, but many of them don't have ligands that are identified because the downstream genes are not identified. They're called hypothetical protein. And so we don't really have an idea of what the metabolite is that could be, that the riboswitches could be binding. However, we do know some of them. And riboswitches are very diverse. They can bind purines, amino acids, metals, and ions. And each riboswitch that binds to the ligand has a unique three-dimensional structure. And what our interest is to understand how the structure of each riboswitch allows us to bind to each of these different ligands. So one of the first projects that I tackled in Dr. Sergenov's laboratory was understanding the 3D structure of the B12 riboswitch. As I mentioned earlier, this was one of the very first riboswitches that was discovered in the early 2000s. And the structure remained elusive for a really long time because of the size of the riboswitch. It's one of the biggest riboswitches that has been discovered, and the chemical instability of the ligand because it's light sensitive. But I was able to solve the riboswitch structure, which I'm showing here, and you can see that there's a large, it's a pretty large and folded riboswitch RNA. It has a four-way junction in the middle with peripheral regions that are making specific contacts to help stabilize the RNA structure. And in the middle of all of this, there's a ligand. This ligand sits in an open binding pocket and is stabilized by interactions that are located around this adenosylcobalamine moiety. And what I didn't mention in the previous slide is adenosylcobalamine is the active form of vitamin B12. So we call it usually the adenosylcobalamine riboswitch. And one of the interesting points about this riboswitch is that while the whole ligand is pretty big, there's an adenosyl moiety that makes non-canonical Watson-Crick base pairs with an adenosine which is part of the riboswitch RNA. What's interesting about this is if the adenosine is mutated to a U, for example, the binding to adenosylcobalamine is greatly reduced, meaning that this adenosine is extremely important in the recognition of this whole large ligand, even though it's only interacting at this one specific region. And besides this important interaction, there's also other interactions that help stabilize the riboswitch that I'm just showing in this schematic here. Other tertiary contacts that make that are important for the stability of the helices of the riboswitch to control gene expression. And so what our work showed from this structure is the mechanism by which the riboswitch is able to regulate gene expression. In the presence of adenosylcobalamine, it sequesters this region. It makes the riboswitch fold in such a way that sequesters this region, that I'm showing here in green, into the riboswitch fold, allowing the terminator downstream to fold and no transcription to take place. However, when there's no adenosylcobalamine present, the riboswitch takes a more open fold, and this green region is able to pair downstream and form the anti-terminator. So there have been a handful of structures that have been solved, and many of the riboswitches, or most of the riboswitches, have very unique structures that differ from one another, that I mentioned earlier. However, there's a very interesting example of the adenine and the guanine riboswitch that recognize chemically similar ligands and fold into a similar structure. These riboswitches were predicted by bioinformatics again, and what researchers noticed was that there were conserved cytosine and uridine residues that they predicted to bind to the ligands guanine and adenine. And indeed, when the structures were solved, we could actually see that cytosine was making, the cytosine of the RNA was making Watson-Crick base pairs with the guanine, and the uridine was making base pairs with the adenine. Thus, specificity for similar ligands can be adjusted by small changes in the ligand binding pocket, and that's what this riboswitch was showing. Recently, there's been a new riboswitch class identified whose ligands were recently elucidated. This riboswitch class was actually discovered back in about 2004. However, we didn't know what the ligands were for a really long time because this riboswitch class controlled many different proteins, and basically the way that you would figure out what the ligands are is by manually testing different ligands. However, last year, there was a publication that came out saying that the YKKC class of riboswitches was able to bind to guanidine. And more recently, two other ligands were identified for this class, PRPP and PPGPP. As you can see from the secondary structure schematic, these riboswitches look fairly similar. However, they bind chemically distinct ligands. There are a couple differences that I want to point out between these riboswitches. The first is in the PRPP and PPGPP structures, there's an extended 5' and 3' region that is not present in the guanidine structure. And the differences between the PRPP and PPGPP riboswitches is even more subtle. And that is that in the PRPP riboswitch, there's a conserved G located here that is not conserved in the PPGPP riboswitch. So I just want to talk briefly about these ligands and what they do. PRPP is a product of the pentose phosphate pathway, and it's the precursor in the purine biosynthesis pathway, where PRPP goes through many changes that are acted upon by these proteins and coded by these genes that are all actually clustered in an operon. And not surprisingly, these genes are regulated by the PRPP riboswitch. And in the end, they make ATP and GTP. The PPGPP ligand, of course, is responsible for the stringent response. It's an important alarmone and is responsible for the stringent response in bacterial cells. The other major difference between the riboswitches is that there's about an 8 angstrom shift inward of the guanidine riboswitch loop in this region, making a tighter binding pocket for this smaller guanidine ligand. So to understand the differences between the two riboswitches, and how they recognize the ligands, we want to take a closer look. First, let's look at the riboswitch from the bottom up. You can see that the ligands sit in a tunnel in the right helical stack. If we look at the RNAs from the top down, we can see that the PRPP riboswitch has this bulged region here that isn't present in the PPGPP riboswitch. This bulge is made by the conserved nucleotide G94 and is making long-distance interactions with a C that's on the opposing strand, and this interaction is used for for stabilizing the structural core of the RNA. This G is absent in the PPGPP riboswitch, and instead the nucleobase of the ligand takes the place of the conserved nucleotide and makes these long-distance interactions in order to stabilize the RNA in a similar fashion. To probe the importance of this G nucleotide in the PRPP structure, I either deleted it or mutated it. And when you do this, and I tested binding by isothermal titration calorimetry. When I did this, I can see that the mutations led to no binding in the PRPP, meaning that this stabilization, this nucleotide is very important for the stability of the RNA. However, when I tried to bind this riboswitch with PPGPP, I could see that there was binding, meaning that the nucleobase of the PPGPP ligand was replacing the conserved nucleotide and restoring the stability effect that is needed for the folding of the RNA. So besides this one important residue, there aren't too many nucleobase changes or differences between the two riboswitches within the ligand binding pocket. And even though the structures look very similar, the way that the ligands are binding are actually quite different. In the PRPP riboswitch, the ligand is making about 10 contacts with the bases within the ligand binding tunnel, and it's mediated by only one magnesium cation. However, in the PPGPP riboswitch, the ligand is making only 4 contacts with nucleobases, and there's many more magnesium-mediated contacts. Finally, to probe the importance of the nucleotides within the tunnel, within the ligand-binding tunnel, I made several mutations to these nucleotides that I'm showing here, and I measured the binding affinity. Most of the mutations that I made resulted in a very big reduction or no binding in the ligand, indicating that these nucleotides are important for the recognition and stability of the RNA. I also performed ribonuclease V1 footprinting to see the structural impacts of these changes on the RNA. And what it showed, and this is like an example of my experiment, but basically the results showed that there weren't many changes throughout the entire RNA when making these mutations. However, there were some reductions in cleavages, or basically protections, that I'm showing here in red when you introduce the PRPP ligand. And what this showed us was that upon the introduction of PRPP, the P1 helix, or this closing helix, is stabilized further, and that's important for the mechanism by which the RNA regulates gene expression. And so finally, what this study showed was that previously bioinformatics had predicted that this was a transcriptional on-riboswitch. And our structural and biochemical work supported these results and actually showed that when the ligand binds, it interacts with the 3' region of the RNA, sequestering these nucleotides from binding to the downstream region, which then allows for the anti-terminator to be formed, which is part of the riboswitch, and genes to be able to be transcribed. However, when there's no ligand present, the terminator is formed and gene transcription is turned off. And so in my talk, I mentioned that riboswitches are a means of quickly controlling bacterial gene expression, and that structural studies provide insights for better characterization of riboswitch consensus sequences. Of course, understanding the structure of the riboswitch guides the rational design of antibacterials, and riboswitches can be tailored to have new specificities in order to create controllable metabolic networks for research and biotechnological applications. And with that, I'd like to thank my lab and my school, NYU, and my funding. Thank you.
Open for questions?
Yes?
How would you determine the analysis of the structure? What methods would you use?
X-ray crystallography.
Would you grow the crystals?
Yes?
And did you also have the messenger RNA without any B12? And you have both types of crystals?
No, actually, riboswitches are much less stable without their ligand, and there's only been a couple riboswitches that have been solved without the ligand bound. So we actually don't have a crystal of no B12 bound to the riboswitch. But for the second project, for the PRPP riboswitch, we actually did get what we call an apo structure without the ligand. But it was actually quite interesting because the last nucleotide of the 5' end had 3 phosphates on it, and it was like pointing in to the ligand binding pocket. So it was like sort of replacing this ligand. So I think, so the structure was actually not very different because I think it was stabilizing the P1 helix. Otherwise, it's like quite difficult to get crystals, so maybe other methods would be better used for that.
Okay, thank you again.
Thank you.